And it's my pleasure to introduce our next speaker, Dr. Richard Steet, who received his Ph.D. from the University of Colorado Boulder in 2000 and completed his postdoctoral studies in the laboratory of Dr. Stuart Kornfeld at WashU at St. Louis University before starting his own lab in 2006. In 2018, he moved to the Greenwood Genetics Center, where he has served as the director of research for the past 8 years. His lab is broadly focused on defining disease mechanisms associated with rare disorders, and the major focus of this research is centered on lysosomal storage disorders. More recently, Dr. Steet has developed new functional platforms for the MPS disorders aimed at helping resolve uncertain variants and uncover new mechanisms of disease. He is engaged in numerous other functional genomics projects at GGC and is now pursuing drug repurposing efforts for multiple rare disorders. He's a strong advocate for rare disorders, and Dr. Steet serves on the scientific advisory boards for numerous family-led foundations, including our own. Thank you.
Thank you, Scott, for that wonderful introduction. My one disclosure, this is not a gene therapy talk. I'm going to take you into a different area of research, one that deals with trying to unravel the sort of complexity and impact of variants that occur in MPS genes and how that's teaching us, or what that's teaching us about how to better diagnose and manage these patients. So I'm going to start really with the first half of the talk for the patients and the families. I'm going to try to explain what I do in the simplest terms, give you a big picture overview. Then admittedly, it's going to get a little sciencey and nerdy towards the back half, but hopefully you'll have the context, I think, to appreciate what we're doing. I'm going to highlight the work on our MPS 1 platform where we've done the majority of the work, but also highlight what we're doing now for MPS 2, and I'll finish with some future directions. Okay, so what do we mean when we say functional resolution of MPS gene variants? What does that involve? Well, let me just kind of make sure to clarify this term. So when I use the term variant, it means the same thing as mutations. These are changes in the DNA sequence that may be associated with disease. We like the term variants better than mutations. How do we find these? Well, clinicians and researchers find variants when they do genetic testing. All of you that have an MPS diagnosis had some form of genetic testing. This might have been an exome sequence or a genome sequence, but that's how these are identified. And you know, why do we end up doing the genetic testing? Well, both genetic and biochemical testing is done because there are concerns for an MPS condition or any rare condition that are noted by your doctor upon the initial exam. We also do genetic testing as part of newborn screening. So if you get a positive newborn screen, and this is in states that are screening for MPS disorders, one of the things that'll happen right away is you'll do some genetic testing and ask, are there variants that might help explain what's happening? So variants that are identified in testing come in different flavors. There are some that we call pathogenic or likely pathogenic. These are ones we know are disease-causing. There are others that are benign or likely benign that we know are not disease-causing. And then there's a large class we call VUS. These are variants of uncertain significance. So these are ones that have an unclear clinical outcome. For some variants that are identified through testing, we already have enough information to be able to say this is the answer, right? Usually because we've seen that variant in an individual that is affected with an MPS disorder. So we have the confidence to say, okay, you can expect that this is what the consequence will be. But there's a huge number that we just have not seen before. So we don't know what the effect is going to be. A lot of those are being identified through newborn screening, and these are the ones that are really causing a lot of problems. The main point that I want to get across is not being able to confidently predict the clinical outcome of someone's genetic testing results is a major problem in this field, and it's a problem for all rare diseases. Why is it a problem? Well, if we can't confirm the diagnosis, it causes all kinds of delays in how to manage these patients. We might not even know, do we need to start treatment? or not, right, if we don't have the confirmed diagnosis. From a family patient perspective, you have uncertain results, that's stressful, causes a lot of worry and concern. So what are we doing? Well, we're not doing this alone, but we and others are just simply trying to do experiments to uncover the functional impact of those MPS gene variants, and then we hope to use that insight to better predict or resolve, that's where the term functional resolution comes in, what the future clinical outcome will be. We're basically in the crystal ball business, right? We want to look ahead and say we can be confident in predicting what the future will be. Now, that future might be one of concern and caution, and one that would require more maybe regular medical monitoring, or it could be one that's more optimistic and less of a concern. That is not Mark Haskins in the middle picture there. That's the evil wizard from Lord of the Rings, but we all know, and by the way, we all know Mark is a good wizard, so just to clarify that. All right. So what do we do? How do we, what does this involve, functional resolution? What are the experiments that we do, and what's the information that we learn? In all the cases that we're doing, we make cell lines that are lacking the different MPS genes. So we get rid of the normal copy of that gene, so it's basically a knockout of that gene. Then we put back in DNA sequences that will make the variant-bearing MPS enzymes. So when we look in these cells, we're only looking at that variant form of the enzyme, and we're not kind of having to muddy through also having the normal copy there. And then we do experiments to try to understand how the variants affect MPS enzyme function. The MPS genes encode for enzymes. You guys have heard a lot of this throughout the meeting. Those enzymes go to the lysosome and they degrade GAGs. So we're asking basic questions. Do the variants lower the activity or stability of the enzyme? Do they cause the enzyme to misfold early in its biosynthesis? In that case, you would have no enzyme activity and it would never reach the lysosome. Or does it cause the enzyme to not reach the lysosome, but maybe it's still made and it gets stuck somewhere? These are the types of basic questions that we're trying to ask. And then we'll take that information and we will use that information. A lot of times we'll pass that information along to these expert panels. These are groups of researchers that gather to try to go through all of the available evidence about a variant and give it a formal classification, make a determination. Is it pathogenic? Is it likely pathogenic? Or does it stay as a VUS? That information that we're providing will make it easier to reclassify those variants. And then if they're reclassified, it's better and easier in the future to predict those outcomes. So I hope that's clear to you. If it's not, please let me know. But hopefully that gives you the context for the rest of the talk that I'm going to go through. So as I mentioned, the bulk of our work started with IDUA and trying to understand those variants. We have a knockout cell line that has no IDUA in it. We put back in vectors by a process called transfection, and then we introduce the variant enzyme into that knockout background. For IDUA, we have to do another slower step, takes a couple weeks, where we do an antibiotic selection. All that does is just remove all of these cells that didn't get the vector and enriches the ones that are there. In some cases, we have to go even further and try to do single cell cloning. I won't get too much into that. But once we have our cell populations, we can start doing experiments. And what we do is just measure basically two simple parameters of the enzyme. We measure its activity. We can do that with a simple fluorescent substrate and enzymatic assay that's well established. And then we measure the amount of the enzyme that's there. We do that with a technique called a Western blot. And then we put those two values together, activity over the amount, and it gives us a single value that we call relative specific activity. That means each variant will have one of those RSA values, relative specific activity values, and then we could sort of see what the whole spectrum looks like. And optimistically, we can start to kind of set ranges. We could say, if you have an RSA value of blank or below, we would call that pathogenic. Now, it's not that simple, but at least that's an initial step for how to sort of functionally stratify these. So this is what this looks like in real life. This was a whole collection of IDUA variants that we did. And there are some encouraging things. So what we included in here are some that we knew were benign and some that we knew were pathogenic. And I think you can appreciate, see where all these benigns are? Here's the wild type. We set that at 100%. We're always comparing the variants to the wild type. So 100%. These ones basically look just like wild type. That makes sense. Those are benign and not disease causing. But look at all these ones over here. So these are the ones that are pathogenic, and they fall below this line. This is on a log scale, so that line is 1% of wild type activity. And it really does a pretty good job, although we have some that pop up above that line. And this region in general is where it's a little harder to interpret. But this is the type of information that is shared with these expert panels and then can be used. If something is a VUS, they can decide, hey, Rich, your evidence looks pretty good. I think we believe that now this is a benign, or maybe we think now it is a pathogenic variant. One little word on this. When we do these Western blots, and this is just using an antibody to detect the protein, we're able to kind of look at what the enzyme looks like. How well is it, how much is there, but also is it processed? A lot of the lysosomal enzymes, when they reach the lysosomal compartment, end up getting processed. They basically get chewed back from either end, and this creates forms that are smaller than the original form. You can see that here in this first lane. You have multiple forms that are showing up. Now we know that processing is lysosomal because if we block the function of enzymes or proteases that are in the lysosome with a drug that basically raises the pH, we shift this back up, and I think you can see that. We're getting a shift back up. So in general, we kind of look at this as the degree of processing will help, it correlates with the efficiency of trafficking. If we don't see any processing, the concern is maybe the enzyme didn't reach the lysosome. If we do see processing, good chance it did get there. And this is this is a sampling of what these look like. And I think you can appreciate, I won't go into the details of this, but there are some that clearly do not process like the wild type. Here's that over here. And then there's some that actually process just like the wild type. So that's added information that we're able to learn. Okay. So what have we done over the last 3+ years? We've characterized over 100 IDUA variants. Almost all of these are reported. And these are the types of things that we've learned. I'll just take you through these at a very high level. The first thing that jumped out is the variant effects are complex. And I'm not sure that our single measure of relative specific activity is capturing the full picture of whether or not they're pathogenic. We learned this from one particular variant that we described in that study, and it sort of spurred us to think about, well, we need maybe other assays beyond just this simple RSA determination. And so we're starting to add secondary assays that might give us a better lens as to whether or not it's pathogenic. We also learned that some of the variants that are benign or clinically benign but do have reduced activity can be a problem if they occur in combination with other variants like that. So we published a study that showed that combinations of clinically benign variants can actually lower the relative specific activity into a range where we would have some concern for attenuated disease. In another study, and this gets in kind of to some personalized medicine approaches, we showed that certain variants have a gain of glycosylation effect. What does that mean? It means they change the sequence and then it causes an extra sugar chain to be added onto the enzyme. In a lot of those cases, the addition of the extra sugar chain causes the enzyme to misfold. So it's having a pathogenic consequence. Well, it turns out that there are a new class of glycosylation inhibitors that will block the addition of that extra sugar chain and actually increase the activity of those pathogenic enzymes up to something that's much closer to the pathogenic range. What I like about this study is it highlights how this whole exercise of learning what the variant effects are can actually inform therapy. It can actually translate from just, run-of-the-mill variant stratification or classification to something that does have downstream therapeutic value. And then in a more recent study, and hopefully this will be published soon, we showed that some IDUA genotypes, so that's the combination of the variants, are actually associated with retinitis pigmentosa. So they're associated with a retinal dystrophy in individuals without syndromic MPS. This is really interesting, right? Is this the first symptom in attenuated disease, some of these individuals are in their 70s, and the only thing they've ever had in their lifetime is night blindness or a retinal dystrophy. That's really interesting. That's kind of telling us there may be this new category of very attenuated MPS patients or patients with IDUA variants that have the system spared except for what's happening in the eye. And I think there's going to be a lot more along those lines. All right, let me shift gears over to MPS II. We had been working on this and have kind of renewed our efforts on this, and we're not the only group. Dr. Dixon's group is also working on trying to understand the impact of MPS II variants with a different but complementary method. And won't it be interesting when we can start to compare notes from those two different methods? It's going to give us really good predictive power. So we do the same thing. We have a knockout line of IDS. Instead of IDUA, it's IDS. The one big advantage with MPS II is that we don't have to go through all those selection steps. We can literally put the DNA vectors into these cells and then right away start doing experiments. That just saves us a lot of time, makes the whole thing go faster. But the workflow is the same. We measure activity and amount, and we get these RSA values. The other big advantage, as you know, for MPS II is that it's usually just a single variant, right? It's one variant, X-linked, and so the interpretation is easier. You don't have to try to say, well, what would the effect of this and this in combination be? We can make a better interpretation because we're just dealing with a single variant. One interesting difference with this enzyme is it processes in a little bit of a different way. So it's made as a precursor, and then in the Golgi, it ends up getting cleaved into two chains. The heavy chain, which contains a lot of the catalytic parts of the enzyme, functions of the enzyme, and then this light chain, much smaller fragment there, this SD2, and that acts as kind of a stabilizer. So these two come back together. There's a lot of lysosomal enzymes that act in this manner. They have a heavy and light chain. They reassociate, and then they do their job in the lysosome. But what you'll notice when I show you the blots is it has a different pattern that's consistent with this processing. So here was our initial analysis. We chose a collection of well-known, well-studied variants and did the same things, and we're getting a good sense. So one thing we've learned with this is some of the enzymes process, right? This one here, S470L, really looks a lot like wild type. It has very good processing, but some of these other ones don't process at all. We included one that was a well-known pathogenic variant. This P86L has essentially no activity. It's made, it doesn't process, but it's essentially a dead enzyme. And we have since now expanded our analysis, so we're able to do these more quickly, and we're learning additional things, right? So here's the larger collection of these. You can see the effects. We have some that are well above that 1% line. Again, whether that's the same for these, we have to do more. There's this one over here, right? This is this, let me see if I can see that. Oh, I can look here. That is A346V. So that's another one that has very poor activity. And that one kind of makes sense because it turns out this alanine at position 346 is right next to one of the active site residues. And so if you change that to a valine, this is a picture of the active site of the enzyme, you're probably crowding the other amino acids in the active site, and that's hurting the activity of the enzyme. So some of these are really starting to make sense. Some we were able to determine have actually really good residual activity. These may be ones that we're less concerned about. We also did a very interesting study with our friends at UNC where we looked at 3 different amino acid changes at the same site. So this arginine 493, we looked at what the effect of adding a proline there would be, a histidine and a cysteine. And there had been some concern. We had a good idea that the proline one was disease-causing. The histidine one, it's kind of still murky, but that cysteine was also kind of up in the air. And so we did our study. This is interesting when you can compare amino acid substitutions at the same site and see how they kind of fall out. So none of them process, but you can tell they have very different effects on activity. This one is completely dead. This one's 32%. That's pretty good. And then this one falls at 1.2. 1.2, if we use that 1% line, would be where we would be thinking this could cause attenuated or very attenuated disease. But we need to do a lot more of these to kind of better set that range. And so we can look at the results that we have. All the ones here in red that are pathogenic, they're well below 1%. In fact, they're below 0.1%. And all the ones that are benign have a pretty good residual activity. But there are some of these that were labeled VUS that I think we should take another look at and maybe feel like they would be benign variants. And another really interesting aspect of this is we can also compare our results with laboratory data. That laboratory data is activity from white cells. It's also looking at the amount of GAGs in urine. And we could start to do those comparisons. And in a lot of these cases, it actually lines up fairly well. Like here, this is that R493 to histidine, 32%. Well, if you look at the lab data, the GAGs are normal and the activity looks really good, right? But you can go through this list and start to line these up, and it does start to make sense. So we can combine the results from our platform with available laboratory data, and I think really start to have confidence in our interpretation. All right, so just want to finish with some future directions. I bring this up every time I ask this question, do we need a functional platform for every MPS enzyme? And I want to change that from a question mark to a exclamation point, right? Yes, the answer is yes, we need a functional platform for every MPS enzyme. Our work is obviously, you see 100 variants for IDUA. I would like to do at least that many for MPS 2. We are currently developing platforms for MPS 3A and 3B. That is an effort that is supported by Cure Sanfilippo. Those are almost ready for use, but why stop there, right? We should have a platform for MPS 4, MPS 6, and so on. And we're really just limited by resources and personnel. We have the know-how to do this, but we need those, put some funding behind it. We're going to be continuing our efforts to look at these gain of glycosylation variants. They're not just found in MPS 1, they're found throughout the MPS disorders. Wouldn't it be interesting if we find some that are potentially amenable to these new glycosylation inhibitors as a therapy? We're going to expand our studies on the retinal dystrophies because I think all the MPS disorders are going to have examples where, it's only the eye, right? And those are ones we want to understand. And then efforts that are ongoing with both Gene Spotlight as well as Cure Sanfilippo are to do drug repurposing screens. When we do these studies, we end up with cell lines that express or make one variant enzyme. That is the perfect tool to then go do some drug repurposing screens and ask, are there drugs that work for that particular variant? It's a precision medicine approach. So that work is ongoing as well. I have a wonderful list of colleagues and collaborators, including the person at the top, Dr. Yu, who has done the bulk of this work and done an amazing job with it. We just keep adding collaborators to this list. I'm excited to work with Dr. Munzer's team at UNC as we expand our efforts on MPS 2. And I'm most grateful to the foundations. All this work on IDUA was initially funded and continues to be supported by the National MPS Society. And I'm grateful for that. That's really what's kept this going. And also all the clinicians who have shared their variants of interest, the ones that are causing them problems, those are the ones we want to study. So I'll stop there. I hope I left enough time for a few questions, and thank you.
And I'll open the floor up for questions. Joe.
Just nice to see this work progressing. Is it possible on your cell lines to actually measure GAGs in addition to enzyme using the LC-MS technology that's available in your institution?
Yeah, so that's a great question. The issue that we have is when we do our experiments, we end up overexpressing the enzymes, sometimes 200 to 500 fold. What happens is even though you have an enzyme that might only work 1% as well as wild type, if there's 500-fold more of it, they're usually, and we've tested this, you end up having enough enzyme that you can't detect GAG storage. That's a limitation with what we're doing. That's actually why what Patty's doing is a complementary approach that will focus on storage and mitigation of storage as the main lens. So I think that's going to be powerful. We're also looking at toning down the amount of expression, bringing it down to where we could actually reliably measure GAGs and have some idea.
Because if you could measure GAG, that would be a really nice adjunct to what you're doing looking at enzyme.
Yes, I agree. The one other thing I'll mention is in the newer projects for 2, we will do this, and for 3A and 3B, in our knockout cell line, we are also incorporating the lyso IP tag, so that in those experiments, we can not only ask what's the activity in the cells, but what is the activity in, residual activity in the lysosome. And I think that's just, again, the more of these you do, the smarter you think about them and go from there.
Questions?
It's not really a question, just a comment. Thank you, Rich. We at Odelia Therapeutics are also exploring this RP, retinitis pigmentosa, and MPS genes, because we have noticed that as well, and we've seen it in MPS 3, where attenuated individuals who have mutations in these genes don't present with classic Sanfilippo, but they are presenting with RP in their late 40s, late 30s into their early 40s. So that's something that we're also looking at.
Mm-hmm.
Related to the retinitis pigmentosa issue, we know in lysosomal storage diseases that misfolded proteins are associated with late onset neurologic disease. That's a clear message from the Parkinson story and carriers for Gaucher. Retinal photoreceptors are exquisitely sensitive to misfolded protein responses, and all of these attenuated alleles are going to have some amino acid change that may contribute to that. That may be part of the story, and certainly in the cases of MPS 3C, where they have not documented any GAG storage with the alleles in that, they have seen retinitis pigmentosa. In addition to the retina being sensitive to these misfolded proteins, exquisitely sensitive to misfolded membrane proteins. I'm not saying that's the only cause, but that's also a potential significant cause of this RP, and it won't be addressed by simply restoring enzyme activity. So I think we need to find an assay to assess that RER stress, as we are also evaluating these alleles, so we don't just chase down one pathophysiological mechanism.
Yeah, no, that's a great point. And I think those could be incorporated. I mean, if it's causing stress, even if it's, in fact, if it's overexpressed and it's misfolding, we should see it even more clearly. So we probably need to think about looking at ER stress markers like BIP and other things and saying, hey, is there some component of that?
Thank you so much for sharing that information. This is going to be so helpful with newborn screening and resolving cases that are inconclusive. How do you envision this working with newborn screening programs, or how do you make that connection? Because I think this is going to be super helpful in those settings.
Yeah, that's an excellent question. I mean, that's where I hope these variant curation expert panels really help provide that formal guidance. You can always ask my opinion, but we may want a larger group of people weighing in on it, right? I mean, I have my feel for these. You do enough of them, you start to, but the actual practical guidance, I think, is a big question, right? You know, someone wants an actual lookup table that they could say, okay, this is how we're going to use this if we found this in a newborn screen, right? Versus just calling me up and saying, I'll get Rich's idea, what he thinks about this, you know?
So I remember when we put in the MPS 2 newborn screening nomination, this was a big concern. And it was a really eye-opening experience to hear Barbara Burton talk about her first cohort of cases of attenuated MPS 2. And I think 75 to 80% of those cases, she had cascade data in families that gave a phenotype. It won't resolve all of them, But if we have the follow-up crew really examining family data for these attenuated alleles, that's going to help give us a really strong picture. There's still going to be a need to ferret out the results from some of the other alleles, but a lot of them will just come from family cascade testing,
Yes,
I hope.
I agree. Maternal uncles, I've kind of, seeing what else is happening.
Hi, Rich. My name's Megan. I think there's 2 microphones flying around here, but I'm over here.
Wait a minute.
Over
Oh,
here.
got it. Okay, thank you.
Kristen had just spoke on kind of my thoughts on it too. I mean, obviously, I feel like this is maybe the first time I'm hearing about it in this setting. My son was found, he had a VUS at birth, and obviously knowing more or being able to know more kind of like leads your treatment options even, and like what you're willing to do and that kind of thing. And I'm just, I'm not a scientist. I'm just trying to like gather from what you're talking about and kind of dumb it down to my level. From what I understand, it's trying to figure out what these variants are. Are they pathogenic? And then what does that mean for that specific variant? Is that correct?
Yeah, it is.
Is there a way I can participate?
Is there a
my
way you
son?
can what?
Like, I don't know if my son's variant is like in this already. I don't know how any of that works or how your research
Yes, well,
starts, but
I'm glad you brought that up. I've had conversations with several parents, and it's like he's the guy that's going to ask you if you know the variant in your kids. But that's kind of because I'm interested. I want to know. Do you know? Have we studied it? Have we already done experiments? If it's one that we think, like one of those gain of glycosylation ones, we would want to know that. I mean, we're still, that's obviously an experimental concept, but it's a possibility in the future that could represent a viable therapeutic avenue, right? So
Right.
I would say yes, we want to have, we need a way that you can share that information. It can be, we can decide, okay, can we study that particular variant? Do we already know something about it? And then I agree. I think the point that was raised is how do we make this practical for everybody? And I think that's a bigger conversation that many of us need to have. How do we translate these findings? How do we make them maximally useful to both families and to those that are going to find them invariably, especially as the newborn screening efforts expand across the country? So.
that was my concern too. It's, if this becomes the new standard, which it seems like I think we're at 50%, at least for MPS 2. And I mean, even myself, like I want to get involved in maybe expanding that so it's 100% of the country. So that's going to be the new problem is, you get this diagnosis, a lot of them are VUS, and then where do I go from here? How do I make decisions? And that would be great information for families coming in at that point.
I agree. Yeah, we need to maybe prioritize those discussions.
Hi, thanks for a great presentation and discussion. You mentioned you studied 100 variants for the IDUA and some for the MPS 2 as well. I'm curious what percentage of variants that accounts for how many total number of variants that we already know for each disease, and how close we are to studying all of them or not, and if the plan is to study all of them,
Yeah.
hopefully.
I think we're making a small dent, but hopefully in time. We're not studying ones that we already know are pathogenic. We did that originally. If the field already says this is a pathogenic variant, we're probably not going to get into that. A lot of our efforts, especially on MPS 1, are in that attenuated range, because those are the ones that are causing people the most concern and stress. Is this really going to be something that's disease-causing or not, right? Those are the ones that are coming to us from the clinical providers that are saying, can you provide any insight on this? Because we're uncertain. So, yeah, we need more people doing this. We need, we're going to keep finding these. The number of potential ones that are out there are in the thousands for both of these genes. So I would say we're making a small dent, but at least we can do these, and as the higher priority ones come to us, those are the ones we can focus on. Yeah.
So I'll just end with another question. I may have missed it, but have you started studying The IDUA variants in combination in single-cell populations?
Yes, well, that was the one study. So that's one of the values of our platform. We don't have to just study one variant. We can add two on the same thing, introduce that. And again, in all those cases where you had combinations of ones that normally we wouldn't worry about and maybe are not even reported, if you have combinations, those can actually drive the activity down lower. And that makes sense. It's like 2 hits to the enzyme. Even though independent, they might not be a problem, they might come together and be additive and drive you down into a range where you should be concerned.
Oh, Terry has a question?
Hey, Rich, I caught Dr. Munzer too outside. I wanted to just share this because when I hear cascade testing, I was at a newborn screening meeting in Taiwan where Dr. Hendricks had presented a case where through the generations, the presentation of the disease was significantly different. And I had asked Dr. Munzer really quickly if, he'd seen this, and we had talked about it a little bit because while we start to gather this information, that's going to be really critical to see where that might happen. The case that was presented by Dr. Hendricks was from a family out of Africa, but the presentation from the generations was very different. Going from something that was attenuated to something that was very severe presently of the child that's living. And so we talk about the cascade testing, but I see that maybe something might start to come out of the data as you start to gather things. I just want to make that comment, and if you've heard anything like that or had concerns about that.
Yeah, I mean, that gets into, so one of the concerns we have is when you get down into a very attenuated range, right? And that kind of borderline, that's where the impact of the genetic background, environmental factors, they start to become much more present, right? That's less of a concern when you have no enzyme activity at all. Those factors kind of wash out, but when you have this residual level, that's where you're going to have differences. You're going to have individuals with the same variants that have very different clinical presentations, and that's going to be due to factors that I think we still don't fully understand, but it is things like their genetic background, their ethnic background, and then things like environmental factors. So it's a good point.
Okay, well, I want to thank Rich again. It was a wonderful presentation.